All right. Thanks for watching. We're going to go over DNA replication today. All right. What is DNA? DNA is a structure. It is built of nucleotides. The nucleotides being adenine, thymine, guanine, and cytosine. It's the basic structure of this chain that holds the nucleotides together, and is also connected to a second chain that runs exactly opposite of the first, and thus we create the double helix model, which is illustrated here in this diagram. The structure of DNA is composed of a structural backbone, which is shown right here as the phosphate and the sugar, often called the phosphate sugar backbone. And we can see that illustrated over here on the right by this backbone. There's two of them since we have the double helix. Between them, we have the nucleotide bases, which are shown on the left here. Those ones are the adenine, cytosine, thymine, and guanine. Also make note here that there is a directionality to DNA where we have what is called the 5' end, which is shown on the carbon here between the phosphate and the sugar, as well as the 3' end, which is shown here on the bottom left of the sugar at the hydroxyl group. And that's going to come in a little bit later as we talk about the directionality of transcription. All right. So the process of DNA replication is illustrated here in this diagram. It has all the bits and pieces. Definitely make note of this and refer back to it as we go over the process. Step 1 begins with a signal being sent for DNA replication to occur, and that will occur in the nucleosome, where all of the DNA is contained in chromosomes. All right, and so here we just have that double helix strand. Step 2 consists of bringing in an enzyme. The enzyme is helicase, and helicase is responsible for breaking the hydrogen bonds that hold each of the DNA bases together. And so you can see him here in the middle as he unzips the DNA strand. And we'll also make note here of the enzyme topoisomerase, which is responsible for unwinding the DNA prior to helicase coming in. We'll talk a little bit about that at the end as well and review what we have there. Step 3. Once the strand is open, we bring in single-stranded binding proteins, which are responsible for essentially stabilizing this unzipped DNA and keeping it from making any new connections or really from just doing anything. And so we see those illustrated here as these gray proteins. All right, and then moving to step 4, we actually begin the replication process by bringing in primase, which is an enzyme that is used to insert RNA primers, or RNA bases that are pretty much the starting line for the replication process, and then they are specific to whatever protein is needed, and whatever protein the DNA strand we're producing is going to go through and produce. Also, while we're on this slide, let's just go over here to the right diagram and look over the replication bubble, which is shown here. We have these replication forks, both on the left and the right. And starting from the middle, we can see on the top left side, we have what's called the leading strand. And this is where we can start talking about the the directionality of DNA replication, where DNA is always replicated from the 5' to the 3' end. And that means that this original strand here on top was originally in the 3' to 5' end. On the bottom here, we have a problem where that if this top one was from reading from the left, 5' to 3', the bottom is going to be 3' to 5', meaning we're going to have to replicate, or we're going to have to add 5' to 3' end, which is opposite of the direction we're actually going in this replication bubble. And so what happens here is we end up having to kind of go back and forth. We have to add, stop to once we get to where we were, go back and add. And so we just end up creating these little fragments, which are actually called Okazaki fragments. We'll talk a little bit more about what happens with them in a moment. Moving on to step 5. We're going to begin talking about the actual replication. And that's where we bring in what is called DNA polymerase 3. And what it does is it hooks on to the RNA primer that was left by the primase and begins adding DNA base pairs, matching the original strand. And here again, we see that leading strand going from the 5' to 3'. And on the bottom, we see the lagging strands, or the Okazaki fragments, going the opposite direction. Step 6, which is our final step for this process, is going to bring us to the last enzymes that we're going to be working with, and that is going to be DNA polymerase 1, also called exonuclease. And what DNA polymerase 1 is responsible for is for going through the chain that was just added, or the new template, or the new replicated strand, and essentially proofreading any of the mistakes it will go ahead and correct. It also goes ahead and pulls off the primer RNA bases we had originally put on and replaces them with the correct DNA bases. So it's kind of the management that comes in and just cleans everything up and makes sure it's all ready to go before it moves on to the next step. All right, and so that's DNA replication, which we're gonna, this is transcription specifically. And here are the enzymes for transcription. Brief overview, we're going to go over them again in depth in case you're doing this for any form of a test prep. Here are some definitions that you might want to have down. So again, we started off with topoisomerase, which went ahead and unwound the DNA and prepped it for helicase to be able to split it. Here again is a diagram of the 3D model of the actual protein in case you're interested. Moving on, we had helicase come in, which again, it broke the hydrogen bonds between the bases of the DNA in order for us to use it as a template. Primase came in and identified where we needed to start reproducing and replicating the DNA strand. It left an RNA primer there. Again, it is important to note that is RNA primer, not DNA primer. All right, we then moved on to DNA polymerase 3, which began the actual process of adding bases. That's the meat of the whole process, is DNA polymerase adding the bases, again in the leading strand and the lagging strand. In case you're interested, here is again a 3D model, if you will, of the enzyme and some of the more specific portions of it, each with their own unique responsibilities in the process of adding those bases. So more than welcome to go ahead and do some research on that in case you're interested. We then ended with DNA polymerase 1, which went ahead and did the proofreading and went ahead and replaced that primer we'd started off with, which was RNA. We needed that to be DNA, so it goes back and fixes that. And then one more we didn't quite mention, but is good to mention here, is ligase. Ligase is responsible for putting back together any of the indiscrepancies of the strand. It's the glue that holds it together. And the one that's most specifically known for is for connecting the Okazaki fragments we mentioned earlier, which again are just broken pieces. And if you're interested in that chemistry behind it, here is a little diagram of the specifics. Again, we're not going to go into too much detail on that, but if you're interested, definitely do some research on that. It's very interesting. And to end, we're going to go over the models of replication, which are essentially, when they were originally studying genetics, they wanted to understand what was causing this replication, and they had 3 different models that they wanted to test and confirm which was which. Messelson and Stahl were the 2 scientists who attempted this. And these were the 3 models that they were interested in: conservative, semi-conservative, and disparative. They did decide officially, and I don't like the word proof, but they proved that it is a semi-conservative method